Welcome to Friday Office Hours. We're really excited for you to join us today. Today we have a presentation focused on our all-by-all phenotype curation workspaces that we have available in the Researcher Workbench. So I'm joined by a few of our all-by-all team members who can be here to answer some questions about the all-by-all dataset, which I know has been very popular. So before we get started, I do have a few quick announcements, and then I'm going to present our speaker, and then we'll get into the presentation. So I just wanted you all to know that next week, the Community Campus Partnerships for Health is hosting a webinar for leveraging the Researcher Workbench. This webinar is going to be hosted with presenters from our Research Academy, and they're going to share some key steps in getting started with the Researcher Workbench and using the data available on the data browser and the Researcher Workbench. So if you're interested, scan the QR code to register and learn more. I also want to announce that applications for the National Alliance of Hispanic Health for the Todos Juntos research grants are due Monday, October 28th. This year, they are going to support up to 12 scholars who are using the All of Us data set to study chronic disease in Hispanic communities. So if you feel that your project and analysis is reflective of this, feel free to apply. The successful applicants will get up to $10,000 for their research project. I also want to announce that Aymone Research Fellowship Program is available as well. So if you are a researcher who uses machine learning models or other tools such as that in that field, they are looking for specific fellowships with you. So you can learn more about Aymone at the link there. I will note that Aymone utilizes the All of Us dataset pretty heavily, and they have produced a lot of publications through their fellowship program. So if you want to get collaborators or get more involved, that would be a great place for you to start. And the last thing is, if you are a Researcher Workbench user, we have opened up our community forum on the User Support Hub. We invite you all to join there to view forum topics, engage with other collaborators or users of the Researcher Workbench, or ask some questions about your project, study design, etc. So you can scan the QR code to get access to it, but you will need to be signed in using your @researchallofus.org account. If you have any questions about using the forum, please email us at support@researchallofus.org. So now I'm going to introduce our speaker. If you've been around us a long time, you all know Aymone. Aymone is one of our data scientists here at the Data and Research Center. She focuses on helping curate that phenotypic data, specifically survey and Fitbit data. She also helped with the All by All project as well, and is a plethora of knowledge to our users. We appreciate Aymone and her contribution to creating these featured workspaces, so I am going to pass it on to her.
Thank you, Sam. Welcome everyone. Thanks for being here. Let me share my screen. And so today we'll be talking about the all-by-all phenotypic data, so the source data that was used for the all-by-all study. And if you're a registered researcher, you do have access to this data as well as all the codes that was used to generate that data. So today we'll go through that. So we have two main things that we're going to do with the objective of helping you understand these phenotypic data that were used for the GWAS and RVAS. And so first, I will go through a general background, a quick overview of what is all by all. And then we'll dive into the specific phenotypes in the curation process. And then I'll show you also the summary notebooks that we have created for these phenotypes. So what is all by all? Essentially, the all by all is a large genetic association study that was conducted across a diverse ancestry and a wide range of important human traits and diseases. So specifically, you have two different association studies were conducted. The GWAS was conducted on common variants as well as on the exome call set. And then the RVAS was conducted on the rare variant for the exome call set. So, and you probably are familiar with the definitions, but I just thought I would put it there. So basically the GWAS is just a research method to compare the DNA of many people, and in order to identify genetic markers associated with a disease or a trait. And typically the GWASs are used for common diseases, but then RVAS, that's for typically rare variants association study. So it's to analyze rare variants in a large sample size and cover some genetic association in complex and rare diseases. So what is the advantage of all bio? Because similar studies have been conducted and are being conducted. It really has to do with the extensive and diverse data that all of us offers in terms of genomics data and phenotypic data. So all of us participants come from a wide range of diverse ancestries, including people groups that are typically underrepresented in biomedical research. So that has a potential to lead to novel discoveries. And with all of us data, you also have an increased statistical power because of the large sample size. So as you may know, our goal is to have about 1 million participants enrolled. And right now, we have about 800,000 participants enrolled. And out of these people, about 400,000 participants have had their data curated. They went through all the consent and all the processes necessary to make everything safe and secure and release that to you, the researchers. And out of these people, there are about 80% or more that are from these UBRs categories, with specifically 45% from racial and ethnic minority groups. And I don't need to tell you that these are all our data sets. You probably have seen these slides many times, but for the genomics data specifically, we have about 245,000 people with whole genome sequences and 312,000 people with genotyping arrays. And then we have survey data, we have physical measurement data, we have EHR data, and then Fitbit data. So this is what the study leveraged. And the study used short read whole genome sequence plus a variety of phenotypic data to conduct the association studies. And that was data for about 250 participants that had whole genome sequence data, and they came from 6 major ancestry groups. And in total, all the phenotypes that we used were about 6,637, coming from the drug exposures, the personal and family health history. We have Phecode and Phecode X conditions, and then lab measurement groups, and then physical measurements. So today, the presentation will focus on talking through these phenotypes. So the results of the all-by-all study were released to you, I believe it was in May 2024, in the control tier. And you have 21 Hill matrix tables with the summary of the summary statistics of the association test, as well as about 36,900,000 individual hate tables with the results of the association test by phenotype. So these are available to you in the control tier buckets, and we have we have some resources to help you with that. Let me go back to the previous slides, because I want to talk about the benefits specifically for researchers. So with the all-by-all result study released to you, you can directly explore the genes or genetic variants that contribute to your phenotype of interest without having to rerun the analysis. You don't have to have prior experience with GWAS or RVAS, and you do save a lot of time and cost. So I got these numbers or these stats from the previous all-by-all office hour that was done, and they ran about 2.7 million jobs. And these are the costs that they incurred per phenotype. So about 22,000 for the genome variant, about 1,700 for the exome variant study, and then 5,600 for the gene level study. And the Sage Cloud platform is very powerful, so it's computationally very powerful, but still this is the cost per phenotype. So you save a lot of money and time by just exploring the results of this analysis. Now, we have put out a lot of resources to help you guys explore the results. So there was a fantastic office hours that was done on May 31st. I encourage you guys to go and look at that. So she gives an overview of the analysis, some of the curation process, and then the tables and how to query them. We have an article on the user super hub that gives, again, an overview of the analysis in the table and how to query it. And then we do have also, you have on GitHub publicly available the code that was used for the downstream analysis. Just keep in mind that currently you cannot reproduce this in the researcher workbench, nor do we recommend that because, like I said, it's very, very expensive. But you can go and look at the code and kind of figure out, and see how the results were generated. So that's helpful. We also have a feature workspace that is fantastic. I look at it a lot. And I can show you how to get there from your workbench. So let me unshare that. And then I'll share this screen. Oops.
Okay, we're gonna go ahead.
Can find share screen. 1 second. Oh, yes. Can you guys see my researcher workbench? Yeah.
Yes.
Yes. Okay. So yeah. So you would have to be logged in, and then if you click on this, and then you go to the featured workspaces, take a second. So you have all our featured workspaces, and the one for querying all bio results is called how to query all bio results and analysis details. And if you go under analysis, you have one that shows you the QC details and one that shows you how to query the result. And I highly recommend that one. And then in a second, I'm going to talk about also the featured workspaces that we will focus on today. And they're also under our tutorial workspaces. And there are 6 of them that focus on explaining how each of the phenotypes were curated. And if you open it, I'm not able to open some of these notebooks because they do have counts less than 20, some show row level data, and this is going to be publicly available. So I just will show you, I have some screenshots in my presentation for you, but when you open a workspace, this is basically what it looks like. You have a README. Please read the README. It has a lot of information, and including the source codes for every table and an index of the phenotypes. And then there's a setup. You don't need to open the setup actually, and you don't need to run anything. These workspaces have been run for you, so you can just look at everything in preview mode, including the summary graphs in those summary files. But if you want to, you are able to. You would need to, there's a cell in the README that shows you how to run the setup, so you don't actually need to open this. You can just run that and then get all the files. And then you have a summary files per phenotype, right? And that specifically focuses on that phenotypes and gives you a graphical summary for the categorical variables, and then for the continuous variables such as physical measurement, that you have things like statistical summaries and box plots and histograms. So we'll talk through that some more. Let me go back to my slides now. Yeah, so the feature workspaces that I showed you shows you how to query the result of an individual phenotype through the Hill table, or how to query the result of a single variant level using the Hill matrix table, and then also using the Hill matrix table, how you can query the result on a gene level for a single ancestry. And then you can query things like number of cases, number of controls, and see the QC process details. So then this is the 6 workspaces that we just saw. Again, you have 6 of them, each one per phenotype. And I just went through this. Yeah, so this is what it looks like when you open one of these workspaces. There are very clear instructions to tell you what to do in the README. And this is where you will access most of the source files. So let's go through what is included in each workspace. Each one has participant level data that are generated as CSV files in the bucket. So you have a phenotype data and the source code for that. You have demographics data, which are used for the demographic characterization, and you have ancestry data and the source code for that. In each of the workspace, you have an index of phenotypes. So you have the concept ID or the concept code, the name and the string name of that concept, and then links to individual notebook summaries. And I'll have screenshots to show you what that looks like. Now, in the article that I mentioned earlier, you have an index that combines all of the phenotypes code for all of the 6 groups in one file. So you don't even have to log in the researcher workbench to kind of see, oh, what is this code associated to? You can go and look it up there. And then again, each workspace has multiple graphical summaries, so one per phenotype of case controls or summaries of continuous variable. Okay, so that's it for the background. And now let's look at each of these phenotypes. Now, I mentioned that each workspace had a demographic and ancestry tables. Those are the same for all of the workspaces. So the code remains the same. So I'll go through those two before we dive into each of the different phenotypes. So the demographics table was simply generated just using a variation of the person table in the workbench to get the age at CDR, the sex at birth, and the race. And from that, we put these ages in bins, and we just save the data as CSV. That's it. So this is not real data, but it looks exactly like that. So you got your age, your sex, your race, and then the age group. For the ancestry, we just grab the ancestry prez file from the genomics bucket. That's all we did. And then we just changed some column names and then saved it as a CSV file. And those are the files that we sent to the all-bio team for the downstream analysis. Okay, so now let's look at the drug phenotypes. So in the all-bio study, we have included about 1,054 drug exposures phenotype. So in the drug curation workspace, you will have access in a README to the drug table. It's called R_drug_ table. And the way that that was queried is that we first queried all participants with any drug exposure concept code using the ATC vocabulary, and those were the cases. So we would assign value true or case to these people. And then we queried all participants with EHR, but that didn't have any of these drug exposures. So those would be our controls. And then we used the PWAS package from the RPWAS package to create the phenotype table. And we did use minimum code counts of 1. That question came up a couple of times about the minimum code count. So for the drug, it was 1. And that's it. And this is what the table looks like. You have a person ID, and then each column is a different Phecode. And each of these Phecode will be a different notebook for the summary. So you will have 1,054 notebooks in that workspace, excluding the README. And no, we generated those automatically. Yeah, nobody has time to write 1,000 notebooks, but this is what you can expect. And in the same README, you have an index table specifically for the drugs. And that includes the concept code, the concept name of the drug, and then a clickable link to the summary notebook related to that specific code. So when you open a notebook summary for a specific code, this is an example for the magnesium compounds. You have an overall summary of case and control by age, by sex at birth, and ancestry. So this is a graph of summary by age at CDL for the magnesium compounds within that summary notebook. And then you also have a more detailed summary by two variables, sex at birth and ancestry, age and ancestry. And when I say sex at birth is like one individual sex at birth values. So you have female sex at birth and ancestry, male sex at birth and ancestry for both case and controls. So it's very detailed. And that's basically what the workspace looks like. Now, for the personal family health history, how we queried the data that was sent to the all by all team is that we queried all participants that completed the survey, and we only kept those who reported having personally the condition. So the PFHH survey asked participants whether they themselves or a family member has a particular condition. We only selected those who said, I myself have this condition. And then people who didn't report having personally that condition were the controls. So Ks were those who had the condition, controls were those who didn't. And again, this is what the CSV table looks like, with each column being a question concept ID. And then we have an index also with a clickable link. And we here have a short name for the question, as well as the long string name and the concept ID. Similar to the drug workspace, we have an overall summary per different demographics and more detailed summaries. Now let's look at the Phecode phenotypes. So for this, we use the Phecode map on the PHIWAS catalog website, and we queried any participant that had any of these Phecodes in the Phecode map, and then we kept those with ICD-9 and ICD codes, and those were our cases. And then the controls were anybody with EHR data that were not in the cases. So that was the controls. And then we did restrict those to sex at birth, male and female. And we had the minimal code counts was 2. And we were able to create the Phecode table. And similarly to the other ones, it's column, person ID, Phecode, and then false or true. And then there's also an index table. And similar, we have overall summaries and detailed summaries for each of the notebooks. So that's about 1,857 notebooks in that workspace. The Phecode X, pretty similar. We got the map for the Phecode X on the GitHub repository. And we did the same thing. We kept those people with ICD-9 or ICD-10 codes, assigned a value of true. And then the cases were those who did not have people with EHR data that didn't have that were not in the cases. And then we did the restriction as well. And then we got the Phecode X table. Oh, yeah. So this is actually a screenshot of the Phecode table. The Phecode X set, Phecode X here. So I forgot to put the right screenshot, but it looks pretty much the same. You have the Phecode X, the description, and the group that the Phecode belongs to. And again, we have summaries in the workspace. So now let's look at the physical measurements. So these will be continuous variables. And how do we get the physical measurements data? We considered 7 physical measurements: the weight, height, waist circumference, hip circumference, heart rate, blood pressure, diastolic, systolic. So we simply queried in all participants with any of these values from the data set, from the measurement table. And again, for any of these things, the full code is in the README. So if you go to the README and scroll to the bottom, you'll see the entire code for all of these, including this one. And the result is here. This is not real data, but this is what it looks like. And that's what we sent to the all-bio team. Now, there's also an index, and this is pretty sure there's only 7 of them, and each one links to that particular physical measurement summary notebook. And for the continuous summary notebook, we have a table that has a bunch of descriptive statistics. We have an histogram and a box plot of the value, and then we do have demographic counts per age, sex, ancestry, and then per sex at birth and ancestry, and age and ancestry. So we also have the overall counts and then the more detailed graphical counts. For the lab measurements, were a little bit different. Yes, we still have the index table that has the concept ID, the name of the lab, and then the summary notebook, as well as the lab group that particular concept ID belongs to. And we have in total 60 lab groups. That doesn't mean 60 concept IDs, because some concept IDs did belong to the same group, so they were queried in the same notebook. But with that, we queried each individual measurement in its own notebook instead of doing it in one notebook and then doing the summary separately. So that's because we did not just query the specific lab of interest, we also performed several transformations on the data. And again, the code for that is available for you in the workspace. You can go look at the Curation pipeline, and this is in general what it looks like. So first we query the lab of interest from the measurement table. So let's say we use concept ID 34501 for blood glucose, and then we have created a bunch of functions in the unifier functions.py for unit harmonization to a standard unit. If you've worked with the all of us at all for any point of time, you may know that the lab data is probably certainly the messiest. So this notebook is actually very useful if you just want maybe a way to clean up your own measurement data, right? So this shows you kind of the process that we used. And so we harmonized the unit to a standard unit, and we did remove outliers, which will depend on each measurement. And if you want to reproduce that, that might depend on your study, on what you consider outlier or not, based on your expertise and knowledge. But then we also have several box plots before and after. There's a lot of visual analysis to see if the data looks good, if we need to further remove more outliers or do some more work. So you got to plot before and after unit harmonization. And then for some, not all, we did drop some sites that after harmonizing and plotting the data looked kind of funky, and we had some data quality concerns. There's also some statistical plots there that can help you figure out how to make these decisions. But then you have a function there to help you drop certain sites if you need to. And you can also play with the outliers boundaries as needed and then remove more. And then the result of that curation process is a participant level statistical summary. And each lab group, basically this is a naming convention, measurement underscore, whatever the concept ID is, .csv. And this is what we sent to the all-bio team. And that's what the summary table looks like. We got the minimum, median, maximum count, and the latest meaning the latest value for that measurement, and then the count, because as you know, all bio is long format, so a person may have multiple values for the same concept ID or the same lab. So we do this gives you the latest one and then the total count, and then the other statistical measures. Yeah, that was duplication. So this is not really, it's hard to see, but this is kind of what it looks like before and after cleaning up the data. You can see how it's much more harmonized, and we reduced the number of units from a gazillion to just one through the process, and it's much easier to work with this data. So yes. So before we end here, it was way shorter than I thought. I wanted to point out a few things based on just the question that we've had from you guys and some inputs. And again, I want to reiterate, even with our resources, please, if you look through them and you think that there's something that may be useful for you to be able to use a resource better, we are always open for your feedback and always want to keep adding and helping you to use those resources. So feel free to look through them and give us a feedback. But I wanted to point out a few things. I'm so sorry about these graphs. They're very hard to see. But if you could be, we had this question before, why does it seem like there's less counts of phenotypes in the results table compared to what's in the curation workspaces? Well, that's because the All Bio team did perform additional processing from the files that we sent them for the GWAS and RVAS. So one thing that they did is that they added 30 random phenotypes. They also added, you can't really see here, but they added 3 custom phenotypes for the physical measurement. So instead of 7, it went up to 10. So they added BMI and it added the waist to hip ratio. And they did some additional manual curation that did reduce the number of phenotypes from the 606 something thousand to about 600 and 500. So that's something to keep in mind. And then even on the sample, on the number sample, they did do some pruning and curation that reduced the number of samples to, they still kept about 90% in most cases, which is pretty good. But that's also something to keep in mind. So that's from the article that I pointed out earlier, what they basically make clear that they kept high quality ancestry group and phenotype pairs. So it was about 8,800. And then those all had number of cases superior or equal to 100. Okay, so that is it for the presentation. And yeah, I hope this was helpful. And let us know if you have any questions.